In this video, we are going to learn about spatial vision. What does the word spatial means? If we check the dictionary, the word spatial basically has the same meaning as space. In that sense, spatial vision crudely means it's visual perception that occurs over an area. Confusing, isn't it? Scientifically, it is the visual ability to resolve or discriminate features. Let's look at how spatial vision is being studied. Sine wave gratings are mathematical graphs used to study spatial vision. These graphs are plotted with the brightness against the position of the targets. The plotted graph will look like a sound wave, with ups and downs over an average brightness level. The sine wave can be studied closely by breaking it down further. One cycle of up and down is called the wavelength. The maximum brightness of the wave is known as the crest, and the minimum brightness of the wave is known as the trough. Wavelength can also be measured as the distance between two crests, or between two troughs. How exactly is this wavelength important in studying the spatial vision? Let's plot out the wavelengths in two gratings. The gratings consist of bars of alternate brightness levels, which coincide with the crests and troughs of the wavelength. The troughs, which indicate the minimum brightness level, coincide with the dark bars, whereas the crests, which indicate the maximum brightness level, coincide with the white bars. If we look close enough, we can actually relate this grating to a letter E from a Snellen chart. This is actually the secret of how our eye is able to recognize that this is a letter E. Let's look at how the image of letter E is being recognized by the retina. Our retina works like a camera sensor for the eye. Each pixel is represented by individual cone cell within the retina. Each cone cell does its job by detecting the brightness level at a particular spot. The cone cells that detect the bright spots turn on and create a neural signal. The cone cells that detect the dark spots turned off and create no response. This mechanism allows the retina to detect gaps by gauging the brightness across the target. However, there is a limit as to how small the gap can be detected by the retina. If the gaps between the gratings are too small for the cone cells to resolve, the eye will see the gratings as a solid dark object due to this limitation. This means that the eye has already met its resolution limit, which coincides to the visual acuity we measure during every eye test.